Good evening, Professor. Today I am going to present the lab research plan for the next 5 months. And here is the summary of all the projects. Project 1 to the project 6. Project 1 is describe the IDC PSA biosensor that is submitted already in the Nature Biotechnology and waiting for the reviewer comments. Project 2 is basically for the AFM biosensor using barium titanium oxide chips for the biosensing measurement. Project 3 is a copper BTC biosensor. Basically, this project is related with the MOF. It's a framework, metal organic framework. So we just treated with the antibody and antigen to check the capacitance value in the presence of copper. And the project 4 is related with the nanopore biosensors. And the fifth one is the nanopillar biosensors, and the sixth one is related with the AD diagnostic applications for the biosensing measurement. So project 1 is already published, so I just move forward to the project 2nd. The project 2nd is divided into two phases. Phase 1 is a without graphene and phase 2 is a with graphene. So phase 1, as my understanding in the previous published paper, mentioned there, like we use the IDC chip and to just immobilize, or the deposition of that compound element is related with the barium titanium oxide on the surface. And so up to this level is already published in the Advanced Functional Material journal, but we move forward to the next step is the biosensor measurement. Our next goal is to measure the biosensing measurement in the presence of the biomolecules, that is antigen, antibody, polymer, graphene, so on. So, and the responsible person is for the chip part is Dr. Daniel and Erickson, and the biosensor part can lead with the three members as myself and the Prashant and the Ryun Sang. So the without graphene phase having a two different kind of cases. First of all, as we all know that barium titanium chip we have only two chips. So first of all we have to check this step in the P3 and the P6 chip, and then we move forward to the barium titanium chip, because first of all we have to check the efficiency or the binding affinity on the surface, and then we use with the barium titanium oxide. And also I have mentioned the time period. The time period is 2 months, and hopefully we will complete this project in 2 months. And also mention here to don't forget to take a picture while experiment. And this one is the phase 2. Phase 2 is basically with the graphene with a 2D material surface on the our chip. And as I previous told, like the responsible person for the chip and the biosensor part. And this with graphene system divided into the three different kind of cases. First case is using P3 chip, because here I have just mentioned the graphene oxide in between the step. And the case 2 also using the P6 chip, and the case 3 is a P3 and a P6 chip for the without using any polymer solution, just directly immobilize the graphene oxide, and then we immobilize the antibody to check the efficiency of the surface or the binding affinity with the graphene of the antibody binding affinity, and then go for the antigen. And then if our result is going positive, then we will check on the barium titanium oxide chip. So it's the project 3 is the copper BTC biosensor. It's a phase 1 is already known, like without graphene. So here I mentioned the instrument and the chemical required, that we already ordered the two chemicals, that is EDC and the PNI polyaniline for our next research. And so it's also divided into two cases. First case is to check on the P3 and a P6 chip. So the case 1 is already done, but we have to check one more time the efficiency the efficiency on the P3 and P6 chip. Here the mention is clearly mentioned the steps and measurement, how we measure and how we put on the surface and how much time it will take to dry, and then we have to check the capacitance value with the LCR meter. The same case is also here, because in the previous cases we use the aptus. We use aptus after washing ethanol and DI water, but in the case 3 and case 4 we using the EDC HCl, because EDC HCl is also act as a cross linker as same as aptus, but the efficiency is more stronger than the aptus. So here I mention first in the place of aptus we use EDC, and then we go for the polymer and the copper BTC for the better binding on the surface. In the case 5 and case 6 is totally different, because we use here the aptus first, and then we go for the pani. It's a polyaniline. It's a polymer. It's a kind of polymer. We already go for the plan. We already planned for the buying this chemical, and we use the pani and the copper BTC, and here in the case 6 we just mix together polyaniline and the copper BTC, and then we put 10 microliter on the surface, and then we will check the capacitance value, is that there is a change or not. That's why I just make a 2 cases for this. And also the same kind of the EDC here, the is same, is a polyaniline copper BTC and the pani mixed together on the surface. It's already I already described the whole cases, so it's not getting so much confusion or so much question arises from our juniors, and if there is something happen, then the Prashant and the Reun Singh is also there to help and to assist them. The phase 2 is the graphene, with the graphene. So that's why I have mentioned the graphene picture in this slide. And so in the case 1 is already the same case as the phase 1, but it's a slightly different to use the graphene oxide in each cases. So we just check with the presence or in the absence of the graphene oxide, whether it going in positive direction, whether it going getting the much more capacitance value or not. The same is already described before, but in the presence of graphene oxide, all the steps. Here the project 4 is the basically nanopore biosensor. It takes 3 to 5 months, and here also the same thing that I have done. Is the phase 1 is the without graphene, and the phase 2 is with graphene. We already purchased the graphene oxide, and hopefully in a 1 week we got that chemical, and then we go for this project. And it's already same thing that we have the AO surface, and then immobilization of the AO layer on the glass, and then we use the directly antibody and antigen. And in the case of graphene sorry, in the case of a graphene, we just before antibody mobilization we use the graphene oxide solution for improving the binding affinity with the substrate, because the graphene is a very friendly chemical and it's very good for the binding in any other with the metal, with the biomolecule. So that's why I'm curious to check the capacitance value. I'm curious to work with the graphene, whether it going is better for our research or not. Here is also the same, also for the nanopillar biosensor case. I have no so much idea about this, but it's already defined without graphene 1. But the phase 2 is with graphene. We just cover all the 6 steps, and then we go for the graphene oxide solution, and then we treat it with the antibody and antigen for the check the capacitance value in terms of refractive measurement, because it's a refractive measurement, and the nanopore biosensor is related with the transmittance measurement, and the project 6 is related with the AD diagnostic measurement. So AD diagnostic applications measurement is already under process. There is no as such clarify the step or the like to describe the project yet, but in the meantime I also check the literature or also consider about the Professor Abhijit proposal, and uh, then we will define the step and the work plan, research plan for this. Thank you, sir.